So today we will understand the KRAS levels and its correlation with the FAS receptors and CD8 plus immune cells in the pancreatic cancer patient. So far in our previous videos we have made a hypothesis based on experimental results and mouse model studies that KRAS expressions levels is inversely proportional to these FAS receptors in the pancreatic cancer cells. And the more FAS receptors are expressed on the tumor cells, then those pancreatic cancer cells has a high chance to interact with the CD8 plus cells that will trigger apoptosis in the pancreatic cancer cells. So in order to check whether this is the same conditions that is going on in the actual pancreatic cancer patients or not, they have decided to check the pancreatic cancer patients data, single cell RNA-seq data of the patients that is available in the public domain. So this is the Disney plot of the pancreatic cancer cell patients data. Here different kind of cells are being distinguished that can be seen here, such as this ductal cell type 2, ductal cell type 1, and these T cells, where the T cells, zoom in view of T cells is being presented on the left hand side, and the ductal 2 and ductal 1 type are being presented on the bottom side. So let us first understand this T cells. So this T cells population has been divided into this 12 clusters, and within these T cells they have checked the presence of the CD3D cells, CD4 cells, CD8A cells, and FAS cells, T cells that has these FAS cell ligands that can interact with these FAS cell receptors. And thereafter, once they find out that these populations are present in the T cells, they check the correlations of these CD8 plus cells and these FAS cell receptors in the different T cells metacluster. And they can see that the CD8 plus cells and the FAS cell receptors are being correlated in terms of their presence in these T cells metaclusters. So this further substantiate their validations that they have found out in the previous experimental studies, that is T cell receptors might interacting with the FAS cell receptors on the immune cells. Later on, what they did is they check this different kind of immune cells in these different populations of these ductal cells, that is type 1 and type 2. They check the presence of this KRAS mutations and these FAS receptors on the tumor cells. So these are the presence of this FAS receptor, KRAS mutations and the FAS receptors, and this is the overlap of this KRAS and FAS. However, so far they cannot distinguish from these figures whether the KRAS mutation, what is the relationship between KRAS mutations and FAS receptors. So they in order to check their relations, they decided to do the statistical analysis in terms of the gene expression value of the this KRAS mutations in the cells where the FAS receptors are found, and they can see that gene expression levels are low. Gene expression KRAS mutations, the KRAS mutations where the low KRAS mutations are found, they can see that the FAS receptors levels are low. FAS receptors expression levels are low. It can be seen here. However, where the KRAS mutations are low, they can see that FAS expression and gene expression levels are high, and this is statistically significant. So this further validates their hypothesis from the that they have developed from the previous experiments that KRAS mutations and FAS receptors are inversely proportional. Later on, what they did is they again check this. Right now they check this about the FAS receptor presence of this KRAS mutations and the FAS receptors, and they also check the presence of FAS receptors in the case of this KRAS mutations, where the KRAS mutations are found, and they can see that the FAS receptors are where the KRAS mutations are the gene expression level of the FAS receptors are high in the case where the KRAS mutations are low, and when the gene expression levels of FAS mutations are FAS mutations are high, they can see that low expression level of this KRAS mutations that can be seen here from this figure. And later on they check the presence of this different uh, immune cells populations in the case of where these KRAS mutations are high and KRAS mutations are low, and they have find out that apart from the CD8 plus T cells, which is statistically significant, all of the other cells, other immune cell types are statistically non-significant. This indicates that it's the CD8 plus T cells that is interacting with these immune cells, not the other CD8 plus T cells that is interacting with the FAS receptors, not the other immune cells. And next they did the percent percent survival analysis with these KRAS mutations, when the KRAS mutations are low and when the KRAS mutations are high, and they can see that with the p-value of 0.0071, when the KRAS mutations are high, the percent survival rates are low, that is around between 2100 days, whereas when the KRAS mutations are low, percent survival rate is quite high. So this further validates their hypothesis that they have developed from the previous experiments, that KRAS expression levels are inversely related to these FAS receptors in the pancreatic cancer cells. And the lower the KRAS expression level, the higher the FAS receptors and higher the probability of the CD8 plus cells to interact with these FAS receptors and trigger apoptosis, and therefore there is a high survival chance of the patients that has this low KRAS mutations. So I hope you got a understanding or further evidence of how this low KRAS mutations has the higher probability of more survival rates compared to the high KRAS mutations in the pancreatic cancer cell patient. If you need to understand more about this bioinformatic analysis in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.